Hello students, today we are going to discuss about last topic of this chapter, and that is infertility. So first of all, let me define what is the infertility. It is the inability to conceive or produce children even after 2 years. Here you have to remember 2 years, even after 2 years of unprotected sexual cohabitation. Okay, so let me repeat what is the definition of infertility. It is the inability to conceive or produce a children even after 2 years of unprotected sexual cohabitation. Even after 2 years. Okay, that is considered as a infertility. The reasons for this may be a physical, maybe both kind of male and female, both physical issues may be there. Okay, congenital, okay, maybe disease having, then drugs, immunological or even psychological. These may be the reason. Okay, and these disorder can be treated in the infertility clinics by using some techniques, and that is called assisted reproductive techniques or a technologies, and that is called ART. So now we are going to discuss about all assisted reproductive technologies or ARTs. Okay, and these technologies used to correct the infertility problems, and these type of technology include these all methods. Okay, so first is a GIFT, that is a gamete intrafallopian transfer, that is called GIFT or a GIFT. Second one is a intra cytoplasmic sperm injection, and that is called ICSI. Okay, artificial insemination, that is AI. Then surrogacy, okay, and in vitro fertilization, that is also called IVF or a test tube baby program. Okay, so these all methods can be used according to the situation, and the condition of a female or a male. So now we will discuss each method in detail. So first of all, let me discuss about IVF, that is a test tube baby program, and that is called in vitro fertilization. So here in this method, ova from the wife or a female or a donor and sperms from the husband or a donor are collected. Here you can see. Okay, so this collection of egg from the female donor and collection of a sperm from the male donor, both will be collected. Maybe from the husband, maybe from the wife. Okay, and maybe if female wife is not capable of producing egg cell at that time, donor will be selected. Okay, sometime husband cannot produce sperm cells, so at that time donor will be selected. So wife or a donor or husband or a donor according to this condition, or according depend on the situation which is over there. Okay, so this will be collected, and after that what happen, it will be induced to form zygote under simulated condition in the laboratory. So this fertilization event will be occur into the laboratory condition outside the body. Okay, and that's why it is written under the simulated condition inside the laboratory. And after fertilization, you know very well, zygote will be formed, and zygote will be allowed to grow sometime. Okay, and it will convert into embryo, and this embryo will be shifted inside the mother, or we can say in the female uterus. Okay, and that's why it is called embryo transfer or ET. Okay, and to be very clear, let me understand more by using this diagram. So this embryo transfer, you know very well. Okay, now what happened? So first of all, egg production stimulated by the hormone in the female. Okay, as a result of that, egg will be produced. Then egg will be retrieved from the ovary. Here you can see egg will be retrieved in the laboratory. Then sperm sample will be collected from the male. Then sperm sample and the egg both will be combined here. Okay, and fertilization event will be induced. Then after fertilization event, whatever the fertilization occur, you know very well, zygote will be formed. Here this zygote will be formed outside the body in the laboratory condition. Okay, and after that zygote formation, the zygote will be transplanted. Okay, itself means zygote itself will be transplanted. So if zygote itself will be transplanted, you know very well, it will be transplanted into fallopian tube. Okay, and zygote itself will be transplanted into the fallopian tube. You know very well, some process are required in the fallopian tube up to the blastula formation. You know very well, because you have learned already in chapter number 3. So if you allow this up to the zygote, and if you transfer zygote only, okay, at that time it must be transferred into the fallopian tube, and such a process of a transport in the fallopian tube after just this stage, okay, 2-cell stage, and that is called ZIFT, or zygote formation. If it is transported in the fallopian tube, that is called zygote intrafallopian transport. Okay, and that is called ZIFT. And if the zygote is undergo certain amount of stages, Okay, and it will reach up to some stages level like more than 8 to 16 cells or a morula stage or a blastula stage. It will be allowed inside the laboratory to grow. And after that, if person is having this type of a transfer, okay, then you can understand this is called intrauterine transfer, because you know this whole process which is required from 2 cell to 16 cell. Okay, inside the fallopian tube it is required 2 cell to 16 cell process. Okay, so if you just transfer these cells at the after zygote formation, then it must be transported into the fallopian tube. And if the person or a doctor allows this zygote to grow up to 16 or more than 16 cell, then it will be transported into the uterus. So if this zygote is transported itself into the fallopian tube, then it is called Zyft. And if it is allowed to grow and after reaching up to the 16 or more than 16 cell, and it will be transported into the uterus, then it is called intrauterine transfer, that is IUT. In both cases, fertilization will occur outside the body. Okay, and that's why it is called IVF or in vitro. In vitro means outside the body fertilization occur, and that's why it is called IVF. And whole process in normal terms we are calling it as a test tube baby. So now let me discuss more about embryo transfer, and that is called ET. As I told you, ET can be of two types. First is a Zyft and second one is a IUT. So first of all, let me discuss about zygote. Okay, you know very well how embryo is formed. First is egg is collected, then male sperm is collected, fertilization event occur in the laboratory, then embryo will be grown. Okay, so first of all, let me see what is a Zyft. So zygote intrafallopian transfer, that is a Zyft. You have to remember all full forms, like, okay, so what is a ET, that is a embryo transfer. Okay, Zyft full form, that is a zygote intrafallopian transfer. Transfer of a zygote or early embryo. Why it is called early embryo? Because it is just allowed to grow in the laboratory up to just 8 blastomeres. Okay, and that is early embryo. So if zygote or early embryo is transported, then it must be in the fallopian tube. Here you can see. Okay, so egg are aspirated or collected in the laboratory and placed in the prepared semen sample, and then fertilization occur in the laboratory, and then these will be transported along with this type of injections into the fallopian tube. Okay, and this type of transfer is called zygote intrafallopian transfer, ZIFT. Next one is a intrauterine transfer, in which you can see transfer of an embryo with more than 8 blastomere. So what you are transferring is matter here. Okay, if it is up to the 8 blastomere, then it is considered as a Zyft, and transfer must be in the fallopian tube. And if more than 8 blastomere is there, then you have to transfer these into the uterus, and that is called intrauterine transfer, and IUT. Here you can see. Okay, whole process will be done in the laboratory and then allowed it up to grow under the certain condition up to more than 8 blastomere, and finally it will be transported into the uterus of the female. Okay, and that is called IUT. Embryos formed by in vivo fertilization. Okay, means sometime it may be inside the female, that is a fertilization within the female body is also used for such a transfer to assist those female who cannot conceive. Sometime what happen, in vivo fertilization will occur in one female, and that one female in vivo fertilization occur, and after zygote formation or blastula formation, that will be shifted, this embryo which is developed will be shifted or transported in another female, okay, which cannot conceive or get pregnancy. So by this way in vivo fertilization also occur, in which fertilization may be occur in the female. Okay, and that embryo will be transferred in the another female. Okay, this is also one kind of a method of in vivo fertilization. Next method is a gamete intrafallopian transfer, and that is called GIFT. So let me understand what is that. Transfer of an ovum. Okay, here you can see ovary is there, and it is responsible for formation of an ovum, and that is from the donor into the fallopian tube of another female. Here what happen, ovum will be accepted as a donated ovum from the another female, and that ovum is transferred in the another female, because this female is not capable of doing this process. Okay, ovum formation is not process in this female. This female is not capable to produce ovum. That's why she need donor ovum. Okay, and by this way transfer of ovum from the donor into the fallopian tube of another female. Here the gamete will be transported into the female of a fallopian tube. Okay, so you must be remember here transportation occur in the female because just gamete is transported, not fertilization means after fertilization event is not there. Okay, so fertilization will occur inside the female body. Okay, not outside the body or not in the laboratory condition, and that's why it will be transported into the female fallopian tube. Okay, so transfer of an ovum from the donor into the fallopian tube of another female who cannot produce ovum but can produce suitable environment for the fertilization, okay, and development. So the when this method is used, you should also remember. Okay, so each method having its own significance. Okay, according to the situation, according to the condition, these methods must be. Next method is the intracytoplasmic sperm injection, and that is ICSI, intracytoplasmic sperm injection. This is nothing but one kind of a micro injection. Okay, we have already learned in biotechnological chapters that what is a micro injection. This process follows same. Okay, so here a laboratory procedure in which a single sperm from male partner is injected directly into the egg cell from the female partner. Okay, and here you can see how it is injected. Okay, this is called micro injection. This is the ovum, and by using this micro injection, the sperm cell directly injected into the ovum. Okay, and after the fertilization, the embryo is implanted into the woman's uterus. This whole process will occur outside the body. Okay, and after this fertilization only, it will be transported, embryo will be transported or implanted into the woman's uterus. Next method is artificial insemination, and that is called AI technique. Okay, so here the sperm will be collected from the husband sometime. Okay, and sometime from the donor if husband is not capable of a spermatogenesis. Okay, so at that time a donor sperm cell will be collected, artificially introduced into the vagina. Here you can see artificially it will introduce into the vagina or the uterus of the female. Okay, by using some instrument it will be injected. Okay, like this. Here you can see it will be inserted directly into the uterus, okay, of the female or the vagina, sometime in the vagina, sometime in the uterus. So artificial insemination into the uterus is known as the intrauterine insemination. If this artificial insertion of a sperm cell occur inside the uterus, then it is called intrauterine insemination, IUI. Okay, and if it is inserted into just a vagina, then it is just considered as a AI or artificial insemination. So depend on where it is inserted. If it is inserted just up to the vagina, then it is called AI, and if it is inserted inside the uterus up to uterus, then it is called intrauterine insemination, IUI. And this technique is useful for the male partner having inability to inseminate female, okay, or a low sperm count. So according to the condition of a patient, this technique must be selected by the doctor. Next method is a surrogacy. Here a woman who is a surrogate mother bears a child for a couple. Suppose this is a couple and this is the woman who is bearing child for this couple. Okay, and she is considered as a surrogate mother. And just because the wife is infertile, sometimes wife may be infertile or unable to carry the pregnancy. So if wife is not able to carry pregnancy or infertile, at that time surrogacy play a very important role. And in this method, surrogate mother is impregnated through artificial insemination or maybe implantation of a embryo. Okay, and that is produced by the IVF, ZIFT, or you know very well IUF. Okay, so by these two methods, this whole process will be carried out. So now let me understand more by using this diagram. What is a surrogate mother? Suppose this is a couple. Okay, husband and wife. Husband can produce sperm cell, but female cannot produce egg cell, or a female may be infertile or cannot able to carry pregnancy. So at that time what happens? Sperm cell will be collected from the husband, egg cell will be collected from the female. Okay, sometimes female is not capable of even producing the egg cell, so at that time donor egg cell will be taken. Okay, so then sperm cell will be collected, egg cell will be collected from the donor or a wife. Okay, then fertilization occur inside the laboratory, and then that fertilized embryo will be transported into the uterus of another female, and this female is now called surrogate mother, and she will carry pregnancy for this couple, and such a phenomena is called surrogacy. So now let us discuss about some problems of ART. So here it will require high precision handling by specialized professional. Very accurately it must be performed, and expensive instrumentation. Okay, and therefore these facilities are available only in very few centers, because it is required so many type of instrumentation, and that is highly expensive, specialized professionals. Okay, and this is requires so much accurate handling. Okay, and that site is available only in the few centers, and here emotional, religious and social problem may also occur. Beyond ART, and to overcome problems related to ART, one more method is available for to be parents. Okay, to be parents, one more option is available, and that is legal adaptation. Okay, and it is one of the best method for the couple looking for the parenthood. So legally, some orphanage is there in the India, in the internationally also some orphanage are there where some child are there who do not have any parents. Okay, and they require parents, and such a parent who do not can get some child, and they will require children. Okay, so legal adaptation is one option, and to getting this option, one can be enjoy the parent. This is your homework for today's lecture, and before we finish this lecture, let me summarize whole topic by using one animation video. So look at this video.
It was mid-1970s. Leslie and John Brown, a Bristol-based couple, were childless even after 9 years of marriage. After visiting scores of doctors, they finally approached Dr. Patrick Steptoe and Dr. Robert Edwards. As an experiment, they would help the couple bear a child. Dr. Steptoe extracted an egg from Leslie. The egg was then mixed with John's sperm for fertilization. The fertilized egg was then placed in a special solution and allowed to divide into 64 cells. The fertilized egg, now an embryo, was placed into Leslie's uterus. 9 months later, on July 25, 1978, Leslie delivered Louise Brown, the world's first test tube baby. Louise's birth was considered a medical triumph and provided hope to many infertile couples who are unable to conceive or produce children even after 2 years of unprotected sexual cohabitation. Infertility can be caused by many factors, including physical and congenital reasons, sexually transmitted diseases, drugs, immunological or even psychological, and lifestyle factors. To treat infertility, couples are often advised to visit infertility clinics, where the doctors evaluate both partners to pinpoint the causes of infertility. Based on the results, the doctors may then suggest medication or surgery. For instance, if a blocked fallopian tube is preventing the woman from conceiving, then the doctor will suggest surgery to unblock it. However, when surgeries or medications fail, doctors may suggest assisted reproductive technologies or ART. ART is a collective term used for clinical procedures that are designed to help achieve pregnancy by allowing the egg and sperm to meet in an artificial environment, usually a laboratory. Today, there are several types of ART available for infertile couples, including in vitro fertilization, intrauterine transfer, zygote intrafallopian transfer, gamete intrafallopian transfer, intra-cytoplasmic sperm injection, and artificial insemination. In vitro fertilization, or IVF, which was used to treat Leslie Brown, is a process in which the egg is fertilized by the sperm outside the woman's body and under simulated conditions created in a laboratory. IVF is also called a test tube baby program. Interestingly, the gametes can be collected from the partner or a donor. Once the egg and sperm are collected, they are placed in a dish for fertilization and incubated together to form an embryo. IVF is followed by embryo transfer or ET, where the embryo is transferred to the uterus using a tube called a catheter. Apart from IVF, couples can also opt for intrauterine transfer or IUT. In this method, embryos formed by fusion of gametes inside a healthy woman's body or in vivo fertilization are removed through surgery and transferred into the uterus of the woman who wishes to conceive. Another option available for infertile couples is Zygote Intrafallopian Transfer or Zift. This process is similar to IVF, where the gametes are collected and the egg is fertilized in the lab. Post-fertilization, when the zygote contains more than 8 blastomeres, the doctors create a small incision in the abdomen using a fiber-thin tube called a laparoscope. The zygotes are then transferred to the fallopian tube through the laparoscope. Interestingly, a laparoscope is also used in another ART called Gamete Intrafallopian Transfer or Gift, in which a healthy egg from a donor is placed inside the fallopian tube of the woman who wishes to conceive. Gift is performed when the female partner has a non-functioning ovary, but her uterus and fallopian tubes are in a healthy condition and capable of providing a conducive environment for fertilization and fetal development. Did you know that at times, a woman may not be able to conceive because of male infertility resulting from low sperm count, low sperm motility, a damaged or missing vas deferens, or the inability to inseminate? In such cases, the couple can opt for intra-cytoplasmic sperm injection or ICSI, in which the doctor injects a single sperm into each ovum, which leads to fertilization. Post-fertilization, the fertilized egg divides to form an embryo, which is planted inside the uterus using a catheter. Apart from ICSI, artificial insemination is another option in which semen, collected from either the male partner or a donor, is introduced into the vagina or uterus by inserting a catheter. Hence, artificial insemination is also called intrauterine insemination or IUI. Artificial insemination, as well as other ARTs, are helping infertile couples realize their dream of becoming parents. However, one should remember that these methods are expensive as they require highly qualified doctors as well as expensive instruments.
In next lecture, we will have chapter 4 revision test 1, and for that you have to prepare exercise 1 from MCQ sheet. Until that, goodbye.